we're going to start. So, my name is Peter Young. I'm president of Société de Chimie Industrielle, but also head of programs, and I'm very, very pleased to be the moderator for today's webcast because it's not only a very exciting topic, but also a very exciting company in terms of the work that they're doing in this space. Before I talk about today's webcast, though, I'd like to just talk a little bit about the organization. First of all, it's 101 years old. I guess there are not that many that last that long, but it has. And it's a nonprofit, and we're really solely dedicated to a couple of purposes. One is to be a great convener to get people together about important topics and to educate our members in the two industries we serve, which is life sciences and chemicals. By the way, we have a long French name, which is Society of Chemical Industry translated, and that's because a couple of years before the North American branch was formed, the French organization, which served Europe, started, and then the US branch got started. So we inherited the French name. But chimie in French is both chemicals and life sciences, as those of you who are linguists know. And so that's been our full core focus now for 101 years. And the key activities are programs like this, which is interviews and speeches by experts in a whole variety of different areas. You know, for example, earlier this month, we had a whole discussion about the plastics challenge in recycling with Mike Wood of Dow Chemical, which is a serious issue. And also we give scholarships, a substantial amount of scholarships to students studying chemistry, biotech, chemical engineering. And we also have a mentor program, and we award the International Palladium Medal Award to someone who's really contributed towards the goals of this organization. So I'm very proud to be part of this organization. I've been on the board for about 25 years, and almost entire money that we raise goes towards scholarships and operations, and no one takes a salary and so forth, which we're all proud of. So part of our events are these events that we have throughout the year, and because of the pandemic, we've gone virtual rather than the ones we've done in person. But to compensate, we've increased the number of webcasts because we feel that it's an important thing to do. So with that, let me talk about today's program. The way we're going to do this is I'll introduce the speaker, Wayne Carter, who's the CEO of TVAX, a leading company in the whole adoptive T-cell therapy. He's going to give a slide presentation to sort of explain the big picture, but also some of the things his company is doing. And then at the end, we'll have sort of a fireside chat for 10, 15 minutes. But we're going to make sure that we leave 10 or 15 minutes at the end to get questions from all of you. And the way that you ask a question is you use either the chat function or the Q&A function. We'll be able to read the questions and we'll answer the ones that come up. So, but to introduce, I don't really believe in, rereading the invitation. So I'm not going to read through all of Wayne's, bio. But why is this topic really important? The reason why is cancer is a serious problem around the world in the United States. And the traditional ways that you try to solve cancer have been, shall we say, very messy, because things like chemotherapy or radiation, although they can help, the reality is you can't specifically just target the cancer cells. So if you have radiation, you're also damaging normal cells around. And chemotherapy, if you know of anyone who's ever gone through chemotherapy, it's a really tough experience in terms of the effect it has on their body. So this new technology, which is called immunotherapy, is really exceptional because the concept is pretty straightforward, which is you use the patient's immune system to attack specifically the tumors and just the tumors. And it's really what people are calling a fifth pillar of cancer treatment. And the really new area is really this emerging technology called adaptive cell transfer. And essentially, you're collecting and using the patient's own immune cells to treat the cancer. And there's several types of adaptive cell transfer, but the one that is most advanced in clinical development is something called CAR T cell therapy. And the most important thing is the technology is going through trying to break through the obstacles to take it from something that works to something that is very cost effective and timely and can apply to a lot of different cancers. And Wayne is going to talk a lot about that. But a number of CAR T cell therapies have been approved in the last couple of years, and there's more to come. So with that, what I'd like to do is turn it over to Wayne Carter, who is the CEO of and president of TVAX Biomedical, which is one of the leading and pioneering companies in this space. Wayne?
Thank you, Peter. I really appreciate it. I'm going to go ahead and share my screen here so that hopefully everyone can see that. Can you see that, Peter?
There we go. Yes, we can see it.
Excellent. So I really appreciate the opportunity to speak today to the Société and obviously the opportunity to have some open dialogue with your members. For disclosure purposes, I am CEO of TVAX Biomedical, which is a company which is actively developing an adoptive T cell therapy. So I may provide some bias in my comments, but certainly I'm happy to answer any of the questions that you may have. So I actually wanted to start our discussion today by talking about COVID, because we have all certainly, we're all familiar with COVID more than we want to be, COVID-19. And certainly we have all heard about the immunity that we may or may not have associated with COVID. And specifically when we talk about that and you hear it in the press, you hear a lot about antibodies. And you hear about, are antibodies protective? We've heard that asymptomatic people generally tend to have lower levels of antibodies than people that have had more serious infections. But there's a lot of data, and obviously this reference that you see here on the screen here, the Nature Reviews Immunology, which was just published a few weeks ago, references specifically the fact that not just antibodies, but B cells and T cells mediate that immunity to COVID. And so as an introduction to, again, talk about tumor immunity, I wanted to sort of refresh, where we are with immunity with COVID, because there's a lot of similarity, there's a lot of overlap. So clearly it's your B cells, which are responsible ultimately for the production of plasma cells, which produce your antibodies, which are able to bind to and opsonize ultimately the protein on the surface of the coronavirus, and then opsonize it so that it can be phagocytized. So that's sort of a normal mechanism that we're well familiar with and what we sort of think of when we think of immunity. But especially when you think of viral immunity, it actually are your T cells, which are really critical in being able to kill and remove intracellular organisms like viruses or parasites. And so those CTLs, cytotoxic T lymphocytes, are absolutely critical. And we've also heard about the vaccines being developed by a variety of different pharmaceutical companies. And so those vaccines, mostly we hear about the level of antibodies that they're producing, which obviously is a biomarker, hopefully, of their success. But it may actually be the T cells, which are even more important than the antibody levels, which may ultimately help us mediate that optimal immunity to COVID-19. So these are things that we still have yet to learn. And certainly as we progress through the weeks and months ahead of us, we will learn more and more about the immunity and what is most important for our protection against COVID. The other things that you hear about, again, and these are relevant to COVID and to cancer, are Treg cells. These are regulatory T cells. These are T cells which are actually responsible for helping regulate your inflammation. Sometimes these get out of control, dysregulation. And when that occurs, we can have a massive release of cytokines. And so you oftentimes hear about these patients that are in ICU that are critically ill, and they'll have massive releases of cytokines. And these are called cytokine storms or cytokine release syndromes. And they're suppressed by steroids and IL-6 inhibitors. And but we have these exact same issues also when it comes to some different treatments and issues associated with T-cell immunity and cancer. And so it's relevant, obviously, to sort of refresh our memory, not only with COVID, but obviously, again, how it relates to T-cell immunity and cancer. And then this is just a final graphic which sort of shows the duration of immune response that we can expect. And so your B cells and your T cells are referred to as memory cells, and T cells in particular have a half-life of 17 years or more. And so they really can provide that long-term immunity to us. And hopefully they will provide that long-term immunity to us with regard to COVID-19. So with that, let me start into the cancer immunotherapy. And so I oftentimes start with this picture. I could probably start with a picture of everybody which is on this webinar today, and then draw a line down the middle of us, because half of us in our lifetime will develop cancer. And so cancer affects all of us, either directly, personally, or through our family members, our colleagues, our friends. And whether they've developed it personally or, we know of somebody that has experienced the effects, as Peter was saying, of chemotherapy, it can be devastating. And it's the reason why people stop their chemotherapy, because they've lost their quality of life. So this is actually a picture of my family growing up. My sister died when she was 50 from a metastatic melanoma. My mom had colon cancer and had significant cognitive effects from the chemo brain, from the chemotherapy that she was on. My dad, who's luckily still alive today, lost half of his jaw from the radiation side effects from treating a salivary gland carcinoma. So in my mind, we still have a long way to go, not only in obviously improving efficacy, but also in safety. And this sort of keeps it front and center for me.
Now, Wayne, are you the one to the left of your mother?
I am.
Okay.
He's laughing out loud.
Well, personalities develop at an early age, right, Wayne?
Yeah. Genetics. So today I'm going to talk about cancer immunotherapy, and specifically I'm going to be focusing most of my time on adoptive T-cell therapies, as Peter mentioned. I will briefly, though, talk about checkpoint inhibitors and cancer vaccines. So this is a slide, an infographic, which really sort of spells out a key problem that we have. And that problem is that in your tumor has figured out how to create a very immunosuppressive microenvironment. Basically, inside your tumor, your immune system is not working properly. So you have immature dendritic cells as opposed to mature dendritic cells. They're not adequately presenting antigen. We have an inadequate production of gamma interferon. We have PD-1 and other interactions which are basically causing checkpoints between T cells and tumor cells and preventing the destruction of the ability of T cells to kill those tumor cells. We have dysregulation of the T regulatory cells. So a variety of mechanisms that exist within your tumor which create an immunosuppressive environment as opposed to what we want, which is really an immunostimulatory environment. And this is really the purpose of these cancer immunotherapies. It is to shift us from an immunostimulatory or a suppressive environment into an immunostimulatory environment. So obviously one with more mature dendritic cells able to present antigen, mature effector T cells able of killing cancer cells, natural killer cells, production of granzyme B, perforin, all sorts of different mechanisms which are able to ultimately kill and keep your cancer not only in check, but actually kill it and remove it from your body. So the first one that I'm going to talk about that I mentioned on that microenvironment slide are obviously checkpoints. And so the most common one, the most well-recognized one is PD-1, which is present on your T cells. And then it interacts with a receptor on your tumor, which is called PD-L1, which is basically the same receptor, which is the ligand for it. When that binds, it basically creates T cell anergy, and it prevents your T cell from working properly, your T cell receptor from working properly, and ultimately resulting in the destruction of that tumor cell. We can block that with inhibitors, and these are generally antibodies. And so there are 3 PD-1s which have been approved. Keytruda is probably the most common, but there are 3 which have all been approved, and 3 PD-L1 inhibitors which have all been approved. And so you can block this, either the PD-1 or the PD-L1 with an antibody, basically break that checkpoint and create then the opportunity for T cell activation and the ability of that T cell to kill your tumor cell. And so that's how checkpoint inhibitors work. And they do work fairly effectively. And they have now been approved for several years and are being used in multiple indications. But as I mentioned, most of what I'm going to be talking about today are adoptive T cell therapies. And Peter mentioned this, and they're literally referred to as living drugs, because we are actually taking your lymphocytes, your T lymphocytes from your body, activating them, expanding them, doing different things to them, sometimes genetically modifying them, and then infusing them back into your body so that those activated effector T cells are capable of killing your tumor. I'm going to talk about 3 today: the CAR T therapy, TILs, which refer to as tumor infiltrating lymphocytes, and vaccine-enhanced adoptive T cell therapy. So the first one I'm going to talk about is certainly the most common and the most well recognized, and those are the CAR T therapies. So they've been approved now for a couple of years. So again, in this situation, the patient exists here. We isolate the mononuclear white blood cells, T cells, via a process called leukapheresis. We take those T cells out of the body, and then we do a T cell transfection, generally with a virus. You can also use electroporation, and essentially put in a new DNA segment into the genome of the T cell, so that then on the outside of the T cell, you then get a new binding domain. And in this case, the new binding domain is specifically CD19. So all of the T cells which have been approved to date, they all target CD19. And these are the 2 which I recognized here, which are the 2 which have been approved to date. Actually, there's another one, I think, which is approved now for mantle cell, but they're all, again, CD19 targeting CAR T therapies. So we basically put the CAR, the chimeric antigen receptor, into the T cell. We expand them and then infuse them back into the patient. And then again, those CAR T cells are then in there to bind to and kill the target. Now, in this case, the target is CD19. And so any cell that is expressing CD19 is going to be killed by these CARs. And so that obviously is good if you have a B cell lymphoma, ALL, whatever. But there are also side effects associated with it. And so here again is the CAR T cell attacking a B cell, the malignant B cell. And so you get tumor cell eradication, which is what you want. But your normal B cells also contain CD19 on the surface of them. And so you will often have a fairly large number of what's referred to as B cell aplasias, where we literally wipe out your normal B cells with this treatment. And so you end up basically with patients with hypogammaglobulinemia. So those patients then need to be treated with gamma globulin to make sure that they're able to stay healthy. So obviously, it's worse to obviously die from the effects of the tumor than having the B cell aplasia. But again, there are significant side effects associated with this treatment. And there are other side effects. One is the cytokine release syndrome. Remember what I mentioned with COVID. Just as with COVID, you can get a massive release of cytokines with CAR T therapies. And so patients can actually die from cytokine release syndrome. We now know fairly well how to treat it, again, with steroids and with IL-6 inhibitors, just like we do with COVID, but it can be pretty significant. And then there are other side effects, neurologic toxicities, allergies, et cetera. But it is these, what we refer to as on-target but off-tumor toxicities that are really problematic for us. And again, Peter mentioned this, whether we're talking about chemotherapy, which has been used for decades, or whether we're talking about new CAR T therapies, we still have these same on target but off tumor toxicities, which really can be quite devastating. So this next slide is a slide which came from Immunologic Reviews about a year ago, which is a really nice slide, which talks about CAR Ts versus TCR receptors, and specifically with regard to the opportunity to attack different proteins. And so, if you will, sort of the tip of the iceberg. And so, the tip of the iceberg is basically the CAR opportunity, where you have basically 27% of your proteome, which are surface membrane-associated proteins. And so, those are the proteins where a CAR could be generated against that target. The TCR, though, your T cell receptor, has the opportunity to actually present any protein, whether intracellular or membrane-associated or surface proteins. Any protein at all can be presented through a TCR receptor. And so the T cell receptor has even potentially greater opportunity for us than CARs do, just because of the breadth of the different receptors and protein receptors or protein targets that we could go after for treatments with a genetically modified TCR receptor. And so there are many TCR receptors which are in development. They, however, also have the same problem. So there have been tissue differentiation antigens and many other types of antigens which have been attempted to be inserted into these genetically modified TCR receptors. And again, we're recognizing that there are a lot of on-target but off-tumor toxicities. So we think we have an antigen, a protein which is specifically related to a tumor. We give it to a patient, and then lo and behold, they develop a neurologic toxicity. And then we do more, of a deep dive, and we realize that protein is actually expressed in the small amounts in particular neurons. And then you have side effects, neurologic side effects, because of that off-target effect. And so again, these I think are going to continue to be problematic for us as we try to develop, whether they're CARs or TCR receptors. It's quite a challenge to think about identifying and developing the right protein and the right antigen to go after to then effectively remove your cancer. There are alternatives to genetically modifying your T cell receptors, and that's what I'm going to talk about next. So your immune system recognizes tumor proteins really in one of three different ways: either mutation of those proteins, and obviously this is obviously evident within the mutations which exist within a tumor cell. You can also just get overexpression of proteins, and that certainly is evident when you think about the B cell lymphomas. We have massive overexpression of CD19 on those B cells, and then we also can have post-translation modification, glycosylation of proteins, which ultimately all three of these mechanisms can result in your immune system being able to recognize them. But it's important to appreciate that there are almost an infinite number of somatic mutations which are possible within tumors. And so the potential list of targets that we could develop is really quite significant. And again, when you think about not only the list of different targets and then the challenge with off-target toxicities, it is really a very significant challenge in front of the biotech industry to try to solve this. And so one way in which we can solve it is by really relying on our own bodies, to our own immune system, to identify and develop the targets, and not for us to try to do them in sort of an artificial manner. And so that's sort of an entree for me to talk about what are referred to as tumor infiltrating lymphocytes. And so this is a photomicrograph of a breast ductal carcinoma. And what you're actually seeing here, all of these small dark dots are actually the nucleus, the lymphocytes, which are present, massive numbers of lymphocytes, T cells, which have infiltrated into this tumor. And so you have to ask yourself a couple of different questions. Okay, so why are all of those lymphocytes there? Well, they're there because your body knows that tumor is foreign and it's trying to remove it. It's trying to respond to it, but it's incapable of doing so. inside actually these carcinomatous cells, you can actually see, some additional few lymphocytes, but not very many, but there are still a few actually inside in really, more interspersed with the carcinomatous cells. But again, those cells are incapable of killing and removing those carcinomatous, those cancerous cells. And they're incapable because of all those mechanisms which I described on that first slide and more. There certainly are other mechanisms of immunosuppression that we don't know of yet. But think now of this particular slide in front of you, and the number of lymphocytes which are present here. They've all been attracted to this tumor for a reason, because they have been activated to one reason or another to go to this tumor and try to remove it. And so one approach to using this technology is a technology known as TILs, or tumor infiltrating lymphocytes. And in this situation, the tumor excises the primary tumor, and then once that tumor comes to the lab, in this case, a company that's developing this is known as Iovance. And once that tumor comes to the lab in Iovance, they actually isolate those lymphocytes from the tumor, and they then culture them, activate, expand those T cells so that they're activated effector T cells, and then they infuse them back into the patient with the goal of obviously, hopefully, killing that tumor, which is still present within the body, because for most of these tumors, obviously, the surgeon is not able to remove or see all of those cells. So the goal here is for the surgeon to remove the primary tumor, and then for these T cells to go in and sort of clean up the rest of the tumor which is remaining. So the reality with TIL treatment is the fact that you have to have a large number of lymphocytes in the tumor to be able to then isolate enough so that you can then adequately culture them and then expand them so that they go into the, and have a chance to treat the patient. So far, the two main lead cancer opportunities that Iovance is moving forward are melanoma and cervical cancer, because both of those cancers are referred to as hot tumors. They have a large number of TILs, tumor infiltrating lymphocytes, in those tumors. And so those two tumors in particular have a pretty good likelihood of being effectively treated with a TIL treatment. But most cancers actually don't have very many infiltrating lymphocytes, and so most cancers are actually going to be inadequately treated by a TIL approach. So that takes me to our approach, and our approach is referred to as a vaccine-enhanced adoptive T-cell therapy. And so in this situation, the surgeon removes the primary tumor, and then we receive that tumor into our lab, and as opposed to isolating the lymphocytes from the tumor, we actually isolate about 50 million of your cancerous cells from that tumor. And then we irradiate those cells so they're no longer capable of growth or replication. And then we take those cells, combine them with an adjuvant like GM-CSF, and then vaccinate the patient against their own tumor. Because most patients actually have an inadequate response to the neoantigens which are present in their tumor. And so this is really putting the neoantigens, all of those surface proteins and intracellular proteins which are abnormal from your cancer, sort of front and center in front of your immune system. So after the vaccination process, we then do a leukapheresis, collect those T cells, just as is done with the CAR T treatment. We take those T cells out of the body, we activate the T cells, expand them, and then infuse them back into the patient, and then follow that up with IL-2 injections. The IL-2 injections actually continue the in vivo expansion and activation of those T cells, and that's done also with the TIL treatment to continue that activation after the TILs, or in this case, the T cells which are infused back in from our treatment, the vaccine adoptive T cell therapy, once they're infused back into the patient. So we believe that this really is a platform technology, and I'll show you some efficacy that we have in multiple cancer types. We refer to this as an autologous cancer cell vaccine-enhanced adoptive T cell therapy, because it is your own cancer. So this vaccine is only going to work for you. It's only going to be relevant to your specific antigens which are present on your own tumor. ultimately we're creating a polyclonal response, immune response to your cancer. One of the great benefits of this is not only do we have efficacy, but we have very few side effects. You know, we've talked about the on-target off-tumor toxicities, and the TIL treatment and our treatment has very few side effects because you're really just getting your own lymphocytes to attack your own tumor. So there are very few side effects associated with it. With ours, you have a little bit of swelling where you get the vaccination, which is what we want, because we want you to have a good, robust immune response. And then you'll have a little bit of malaise and fever for about a day or two in about 1/3 of the patients. So really very few side effects in comparison to losing your hair and developing chemo brain and losing taste sensation and having all sorts of terrible side effects with most of our chemotherapies which are available today. We've also just received FDA Fast Track designation for our phase 2b study, which I'll discuss in a few more slides. So the first data slide that I want to show you is actually not from TVAX, but from one of our subsidiary companies, which is known as Elias Animal Health. So about 6 or 7 years ago, we started Elias Animal Health as an opportunity to develop this same treatment, same technology for the treatment of cancer in dogs. So these are not a cancer model. These are dogs that are client-owned. They're owned by people just like you and me that develop cancer naturally. And so these dogs developed bone cancer, osteosarcoma, which is a terrible diagnosis. By itself, it's a death sentence, usually within a month or two. You can amputate the primary tumor and you can extend survival up to about 134 days, about 4 months. You can do amputation and then aggressive chemotherapy and extend survival up to about 300 days. In this case, Elias did amputation and then the immunotherapy. And so what I want to do is I'm going to blow up this Kaplan-Meier curve so you get a better idea of exactly what I'm talking about. And you can see a little bit better the benefit here. So if you're not familiar with a Kaplan-Meier curve, this is a Kaplan-Meier curve. At time point 0, the survival is 100% because everybody is alive on the first day of the study. And then as time progresses on to the right, you'll see the curve drop down, obviously where a patient dies. In this case, the patient was a dog. And so you can see that we had obviously some drop off pretty quickly within within a little over 200 days of about 4 of the dogs. But what you can see here is, first of all, this dotted line representing the median survival of amputation alone. And so this is where the dogs with amputation, should have survived. So if there was no benefit of the immunotherapy, you would have basically seen everybody around that line. But instead, what you see is a significant impact of survival, which is demonstrating really that clinical benefit of the immunotherapy. And so this was a 2-year study. Half of the dogs, as you can see from this graph, were actually alive at the 2-year study and out well past that 2-year study. And so it's really quite a remarkable effect that simply has never been seen before in osteosarcoma in dogs. And so quite remarkable data and really translational data because what this enabled us to do and identify was the fact of what's the benefit of this immunotherapy when patients, in this case dogs, had not been previously immunosuppressed by multiple rounds of chemotherapy and radiation therapy, because these dogs had never been treated before with chemotherapy. Now, all of the human studies that I'm about ready to show you, all of those patients, even though we did have efficacy, but they also had been very much immunosuppressed by the effects of previous rounds of chemotherapy and radiation therapy. And so this is one of the human studies. This is a graph which again shows a Kaplan-Meier curve, and these are historic controls, and this shows you how quickly people die from brain cancer. So it's obviously terminal disease, and the vast majority die off very quickly. In this particular study, this was actually 2 studies, a total of 43 different patients. And as you can see here in this blue line, you can see we were able to shift that survival curve to the right significantly by over 2x. And so we were able to significantly improve the survival of pretty much almost everybody in that study. But importantly, at the bottom here, you can see that it flattens out basically at about the 20% level. And so 20% of the patients had really long, durable responses to this. And again, these are recurrent brain cancers. So these patients had already had at least one surgery prior to receiving this surgery and then the immunotherapy. They received previous chemotherapy, radiation therapy. And so they not only, were sort of at the end of their life just from the cancer itself, but they also had very suppressed immune systems. And even in spite of that, we were able to see some very significant efficacy here. This is one of the patients. And so this is an MRI of one of the patients. You can see here 3 slices through the brain, and the white area is actually the tumor, the glioblastoma, high grade. I think this was a grade 3 glioma, which was present on 3 different slices here. The enhancing area is that tumor itself. So this is 3 months after surgery and just prior to the start of the immunotherapy. And then 2 months after the immunotherapy, you can see that that tumor has decreased significantly in size on all 3 slices. 8 months later, still present, but just barely so. Ultimately, this was a complete responder that lived over 5 years. So a really remarkable finding, especially considering this is brain cancer, incurable brain cancer, and in a patient that was literally at the end of their life.
I want to inject one comment for those of you who may not be as familiar with glioblastoma. It is quite famous because people who get it die fairly quickly, but the other is Bo Biden and John McClane were both they died of glioblastoma. So some very famous people, sadly, were killed by this. So it's it's a really deadly brain cancer, and anything you could do to solve this problem is a huge benefit.
Yeah, no, I could not agree more. We've also, in our clinical trials, we've also treated 6 different pediatrics. And you can see here, this is a glioblastoma present in a child. And this is before the immunotherapy. And then here again, 3 months after, you can see that tumor has decreased significantly in size. So in addition to high-grade gliomas, we also treated stage 4 kidney cancer, renal cell carcinoma. And again, just like you saw with the other Kaplan-Meier curve, the black line represents the control group. And you can see again, it's a terminal disease. They die very rapidly. With the immunotherapy, we're able to shift that curve to the right by over 2x. And again, we had significant durable responses, in this case, about 1/3 of the patients. So moving forward, we're planning on a phase 2b study in glioblastoma multiforme. This is GBM. This is the worst type of brain cancer that you can have. As Peter mentioned, it's what Bo Biden and McClane and Ted Kennedy, many people have unfortunately succumbed from this terrible disease, and they die very quickly from it. We have received fast track designation for GBM by the FDA. We also have an orphan disease indication granted by the FDA. We're actually going after the worst kind of GBM, if there is even a worse kind, and it's what's referred to as MGM negative, which is the type which simply does not respond at all to temozolomide or radiation therapy. It's about 60% of the patients, and so those are the patients which we're going to be targeting for this study. The market for our therapy, we believe, is actually quite significant, because hopefully, as you've been able to appreciate, we really believe this is a platform technology that can be used to treat any cancer, because the premise behind the therapy is obviously very personalized. It's an autologous vaccine against your own tumor, and then it's your own T cells directed against, obviously, your own tumor. And so it's a very personalized approach, but we believe it actually will evolve into a platform technology that can be used to treat any patient's cancer, and not only treat it, but obviously treat it with very few side effects. So we are targeting other cancers, including pancreatic cancer and biliary tract cancer, for SBIR grants with the NIH. And just to wrap things up, I think it's also important to appreciate that there's I think a very good opportunity here also for combination therapy. So obviously in the canine study, in TIL studies, in our studies, we're at 50% long-term survival or durable, significant durable survivals. And so, but how can we extend that further? And I think one of the ways in which we can do that is by combination therapy. And so this slide in particular is really just a graphic talking about the potential to combine these neoantigen enhanced specific adoptive T cell therapies like TIL or like our vaccine enhanced adoptive T cell therapy, and combine them with a checkpoint inhibitor because as valuable and as beneficial as these T cell adoptive therapies are in and of themselves, they would likely also benefit from a checkpoint blockade at the same time. And so there are certainly a variety of different approaches which different companies are working on. Iovance is combining checkpoints with their TIL treatment, but there are also even some CAR and TCR approaches which are actually combining a checkpoint blockade into their CAR approaches. So just to wrap things up, I'd like to, obviously there are hundreds of people which have been responsible for conducting these clinical trials. Obviously, I'm so appreciative of the patients that have offered to be in our clinical trials and helped us learn more about the technology. But on this slide is obviously Gary Wood, who is the inventor of the TVAX immunotherapy and the founder of TVAX Biomedical. He is my chief scientific officer and just an amazing scientist and a great person. And I close with this slide to again remind us all that not only do we need better, more effective and safer treatments, but the patients are waiting. And we must continue to move forward as quickly as possible to make sure that we can bring an end to these terrible diseases that so many of us are afflicted.
Well, I have a couple of observations and questions, but clearly from that slide, Wayne, no one, if you were running for office, no one would call you low energy Wayne, right? So if you can, let's see.
Stop share.
Okay, yeah, stop share so we can see the bigger picture. So, it's very interesting, when you, over the years, I've observed different breakthroughs in all sorts of things, whether it's treatments or antibiotics or whatever it is. And in the early phase, it's always tough to get all the things to line up, right? And actually, if you go back, I remember when they first, they cracked the human genome and so forth. Well, everybody got too excited, but in fact, it took a number of years before they could figure out then what could you do with that to benefit mankind. I kind of feel that this whole CAR T, T cell, and adoptive T cell is one of those things where 5-10 years from now, we'll look back and say, it's just amazing what impact these things have. However, each treatment has its hurdles, right? And the interesting thing, and I'd like you to comment, is, at least from my understanding in this area, there are two hurdles. One is to get a particular technology to work effectively, but there's also just a cost issue, right? Because you have to extract T cells, you have to treat them, so forth. And so that's why some of the ones that have been, famous ones that have been proved, are very expensive, right? Because it takes them 5 to 7 weeks, right, to process and enhance and so forth. So I think there are two hurdles, and actually Peter Marks, who spoke at a conference last year, he said, it's as critical to solve the cost issue as it is the efficacy and safety issue. And I know you've been really trying to you're breaking through on both, right? Because your approach also attacks the cost issue. But could you comment about those two hurdles? Because they're really, I think, the two key things that will determine whether the use of these technologies will become widespread, which we're hoping is true, right?
Yeah, no, you're absolutely right. And no, they're very good questions. And I think cost is absolutely critical. Again, the ones which have sort of paved the way, certainly our CAR Ts are the ones which have paved the way ultimately now for TILs and for our therapy. They are terribly expensive. But as manufacturing improves and the automation manufacturing techniques improve, I absolutely believe that cost is going to come down. Mm-hmm. And I'm absolutely convinced that, I know that our therapy can be produced and manufactured far less expensively than the CAR Ts. but obviously, we have a long way to go to ultimately prove the efficacy, for a regulatory approval for our therapy, and then make sure that we have the right manufacturing methods in place to most cost effectively, deliver that treatment to the patients. But you're absolutely right. Cost is a critical element that has to be, considered. The amount of money that we continue to spend for cancer treatments and obviously gene therapy treatments and other things, it is exceeding almost exponentially, and we have to get it under control to be able to control adequately our healthcare costs. And as all of us age, we're going to be exposed to cancer more and more, and so we certainly must have cost-effective treatments to make sure that we're able to, effectively manage the hurdles ahead of us in healthcare.
Yeah. One of the interesting things is, like in your case, your technology has broad applications. Now, it happens to be that you're focused right now on a certain area, right? As you should, right? The famous saying is, it's better to do one thing really well before you move on to the next one than to do 3 or 4 things poorly. And so that's what you're doing. But it's clear that your technology could apply to a variety of cancers. Is that true for a lot of the other companies? There are a lot of companies trying to use this general technology, and I guess, are they generally universal technologies, or are they more using technology that's specific to certain kinds of cancers?
So, we really believe that is one of the significant advantages of our approach, because we do believe it obviously it's very personalized. And so you can't just take it off the shelf and inject it into a patient. It's a very personalized approach, but we also believe that that's going to yield a lot of benefits ultimately, for the patient. And so it is certainly different in that regard than other therapies, which are certainly in development or on the market. A lot of other therapies are very specialized. I mean, obviously the CAR T treatments are very specific for a patient that has a tumor which is expressing CD19. If you're not expressing CD19, which is basically a B-cell lymphoma or leukemia, then that treatment is not going to work. And so obviously the CAR Ts, the TCRs, most of those are very specific and very focused treatments. But again, we believe that ours is going to be, we believe and we hope that it's going to be actually tumor agnostic, where it really can potentially treat any cancer which is presented.
Yeah, that's right. If you let's sort of put yourself in a time capsule and move ahead, or a time machine 5-10 years out, what do you think it's going to look like in the whole area of CAR T cell cancer cell therapy?
Well, so that's a great question. I do believe that there are going to be a lot of other TCRs and CAR Ts, which ultimately, it's hard to imagine today, with all of the toxicity that those are going to be, that they're going to be able to easily to overcome that. And yet, knowing how the innovation that exists within our industry, I do think that there will be, certainly a significantly increased number of CAR Ts and TCR treatment approaches. But I also really believe strongly in our own treatment and also the treatment of using the TILs, because in both of these treatment approaches, you're not genetically modifying your T cells. You're using your own immune system to activate and activate those in such a way that the antigens are presented, and then the T cells ultimately are produced to recognize those antigens which are present from your own tumor. And so I think there's going to be a key opportunity to utilize technologies which don't have all of these off-target effects, because ultimately, when you also think about healthcare costs, it's not just the cost of the treatment that you're administering, but it's also the downstream healthcare costs of then managing all of those side effects, sometimes for years or the lifetime of a patient. And so if we can avoid that by using a much and sometimes simpler approach, which doesn't require genetic modification of the T cell, then obviously that's a better approach. So I do believe that the adoptive T cell therapies, especially the ones which utilize your innate TCRs, I think are going to be very effective in not only bringing about efficacy, but also preventing those off-target effects.
Before I go to cancers, I just want to make one observation, and that is the cancer that you're going after is a particularly nasty form of cancer. And having actually, I remember reading about what happened to John McCain, because there was a lot of lot of news coverage, right, of what he was going through, and it was very sad that they said once you're diagnosed with it, if you live more than 14 months, that's more than that, that's unusual. And I remember one point he just said, "I'm going to stop treatment because what's the point, right? What's the point of suffering? Why stop the treatment because I'm not going to live that long?" But I also gather that the type of glioblastoma is particularly nasty because it's really hard to remove it through surgery, right? Because of the very nature of it, right? And maybe you could describe that, because it's a particularly nasty form of brain cancer.
Yeah, so GBM, glioblastoma, is unique in that regard. It does not generally metastasize, but it does send these long finger-like projections through the brain. And so even with very sophisticated image-guided surgeries, which are done today, the surgeon has the ability to remove a large amount of tissue, but can't remove all of those fingers of the tumor which exists throughout the brain. And again, I think that's one of the advantages of our therapy. I don't think it's really best designed to actually remove a massive tumor. I showed that one MR image where it actually reduced that entire image, that entire tumor. But I think that the therapy is actually best designed to treat what we refer to as minimal residual disease. And so that's a situation where the primary tumor has been removed, but we still know that there are either small metastatic tumors or these finger-like projections with GBM that exist in the body or in the brain with GBM. And then those T cells, you can think of them literally almost like 50 billion smart bombs. They've been primed against your own tumor antigens, and we inject them back into your body, and they hunt down your tumor cells wherever they are, even in the brain, and then destroy those cells and hopefully bring about a complete cure for you.
Now we have a wonderful question here from David Hurwitz, who's in the audience, and I particularly like it because the word Star Trek is in the question. So, so I'm getting you all excited about answering this question. So David says, as this is a personalized individual treatment, how long does it take to turn around the vaccine, quote unquote? What is the timing? Thus, how critical is the time window between the time when the patient is identified, I guess presumably with the disease, and they get the treatment? And then the reference to Star Trek was, only on Star Trek are these treatments delivered in the course of a 1-hour episode. So David wins the award for best Q&A question for the year.
So we actually can turn our vaccine around quite quickly, usually within about 48 hours. So we receive it in the lab. We very quickly have a process to isolate the cells, irradiate them, and then combine it with GM-CSF, freeze those cells down, and then prepare it into a vaccine and then ship it back to the site. So it's about a 48-hour process, which actually works very well. So in the case of the glioblastoma, the surgeon removes a primary tumor. We receive that certainly within a day, and the site will receive that vaccine back within about, obviously 3 or 4 days after the tumor's been removed. We generally don't start vaccination until about 7 days after surgery to make sure that the immune system has had an adequate time to recover, begin recovering after that surgery. But yeah, no, we can turn the vaccine around very quickly. We also can turn around the T cells quite quickly. So we actually have 2 different products for our therapy. The one obviously is the vaccine. The second product is the T cells. And we can, once we receive the T cells by the leukapheresis, we receive those, and generally takes about 7 to 8 days for us to activate and expand those cells, and then ship them back to the site so they can be infused into the patient. Now it depends, with the timing, again, with regard to when we actually give those T cells, because we have to, align that, especially with brain cancer, with their standard of care, which is the chemoradiation therapy. But we can actually, from a manufacturing perspective, we can turn them around quite quickly.
So actually, you are like the Star Trek, but that's not true for the other approaches, which are more like the full version of Gone with the Wind, right?
You're absolutely right. You know, the TIL treatments, sometimes manufacturing can take 6, 8, 10 weeks to adequately expand those cells. And the same with CAR T. Sometimes it takes a long time for the entire manufacturing process. So we actually, and that's one of the reasons why we believe we're going to be cost effective, because we can turn around those T cells really quite quickly, which is one of the most expensive parts of the manufacturing process.
We have time for one more question from Billy, and the question is, what does the process look like for CNS B-cell lymphoma, and how do we contact you for clinical trials? So very specific questions here.
Yeah, so that's extremely specific. So if you have a CNS B-cell lymphoma, obviously that would not qualify right now for our or somebody that has one, that would not qualify for our treatment. We're going to be specifically going after GBM. If it is a B-cell lymphoma, I mean, honestly, I don't know why a CAR T approach would not be tried for something like that, because a CAR T approach, obviously, if the tumor is expressing CD19, should work relatively well for that. So I think that would be my response there.
Well, we've reached the end of the hour, and I hope all of you who are in the audience agree that this has been a very exciting webcast. One, I think we learned a lot about a very important area of new science that I personally believe is going to be the whole breakthrough area for cancer treatment. And obviously we learned also from Wayne, not only a good tutorial about what's happening in the field generally and what some of the issues are, but also some of the exciting things that TVAX, his company, is doing. I hope all of you really enjoyed it. You had some really good questions. The fellow who talked about Star Trek, you're welcome to attend any webcast, given the way you ask questions. And but what we really try to do here is to have webcasts in life sciences and in chemicals where really important issues in the industry, whether it's advances in products or industry dynamics or problems that are being dealt with, are addressed and discussed. So I want to thank all of you who've attended. I want to give a particular thanks to Wayne Carter for taking this time to help us understand these developments more. And I encourage all of you to continue to attend Société de Chimie Industrielle events. We have 2 or 3 a month. And of course, I encourage you to be a member and get some of the member benefits as well. So thank you all, and thank you, Wayne. This is being recorded, and so it will be available on the website if it's like Netflix and you want to watch it over and over again, or more importantly, if you have someone you think would benefit from watching the recording. So thank you very much, and thank you, Wayne, and we look forward to, I guess, quote, seeing you, unquote, all of you at future events.
Thank you, Peter.
Mm-hmm.